At 1876, Dr. Robert Koch, a German physician and one of the founding fathers of bacteriology, cultivated anthrax organisms on microscope slides to demonstrate their growth. Throughout the process, Dr. Koch discovered translucent ovals growing within the cell, and that it could remain viable for years. Even exposed under harsh conditions, these ovals will soon to be known as bacterial spores. Naturally, in biology, organisms has the capability to have different coping mechanisms in order to survive, thanks to evolution. For example, a turtle hides in its shell for protection, camouflage, and a help for it to swim faster. A squid will squirt out ink to scare or distract predators so they can escape before being seen or eaten. And as we know, evolution is applied to even the teeny tiniest organisms. That's our case for bacterial spores, mostly known to be seen in firmicutes at the genera of Bacillus and Clostridium species. It is one of the most common coping mechanisms for bacteria. Bacteria form spores to protect themselves against ecological degrading agents such as extreme temperature, radiation, antibiotics, and desiccation. It is the most dormant form of bacteria since they exhibit minimal metabolism and respiration as well as reduced enzyme production. Getting rid of bacterial spores requires sterilization. Sterilization is a process of killing all microorganisms present in a given area or object. One of the most common procedures of this would be autoclaving, but it can also be done via the use of high-energy radiation like gamma rays and chemical sterilants such as hydrogen peroxide. With that said, we can now envision how much of a problem this would pose, as it seems to be totally indestructible, since even our number one weapon against bacteria, antibiotics, doesn't fit the bill. Bacterial spores are associated with several serious diseases, as some pathogenic bacteria can form spores to survive in harsh conditions such as tetanus is an acute, often fatal disease caused by toxins produced by Clostridium tetani. It is characterized by generalized rigidity and convulsive spasms of skeletal muscles. The muscle stiffness usually begins in the jaw and neck and then becomes generalized. It is commonly found in soil, house dust, and animal and human feces. Transition happens when spores enter the body through wounds or cuts. Botulism is a rare serious illness caused by a toxin that attacks the body's nerves. Botulism causes difficulty breathing, muscle paralysis, and even death. Caused by botulinum toxin, which also happens to be used to produce Botox, mainly came from Clostridium botulinum or other related species. It is commonly found in soil and untreated water throughout the world. Transmission would be ingestion of spores or preformed toxins in contaminated food, wound infection, or intestinal colonization in infants. Known to be a common war disease, it is a highly lethal infection of soft tissue caused by Clostridium species, with Clostridium perfringens being the most common. This is synonymous with myonecrosis and is characterized by a rapidly progressive gangrene of the injured tissues along with the production of foul-smelling gas. Infected tissues would turn black and are non-reversible. It is often found in soil, sediments, and areas subject to human and animal feces. Bacterial spores enter the body through wounds, particularly traumatic or surgical wounds. Now that we know that bacterial spores are A. extremely durable and strong, and B. can cause serious life-threatening disease, you probably know where this is going. In 2001, anthrax attacks in America, also known as Ameritrax, took place. It involved a series of letters containing anthrax spores mailed to the news offices and to U.S. senators, just shortly after the 9/11 attacks. This resulted in 22 infections and 5 deaths due to inhalational anthrax. Inhalational anthrax begins with flu-like symptoms, such as cough, fever, and muscle aches. These symptoms may last 2 to 3 days and then appear to go away for 2 to 3 days. Then the illness can come back, resulting in severe lung problems, difficulty breathing, and shock. The attacks had a profound impact on various aspects of American society, public health, and national security. These attacks led to a widespread fear and anxiety, prompting changes in how mail is handled and screened for potential biological threats. The US government and postal services implemented more stringent security measures, including enhanced surveillance and detection systems. The public health response highlighted significant gaps in preparedness, resulting in increased funding and resource for biodefense and emergency response capabilities. The attacks also influenced research and development in fields like microbiology, epidemiology, and forensic science, emphasizing the need for rapid identification and containment of biological threats. But bacterial spores don't necessarily always equate to bad things. As we know in science, once explored, anything could be a double-edged sword. While bacterial spores are often associated with diseases and contamination, they also have several beneficial uses for humans, particularly in medicine, agriculture, and biotechnology. Some spore-forming bacteria, such as Bacillus subtilis and Bacillus coagulans, are used as probiotics since they can survive the acidic environment of the stomach and reach the intestines, where they can promote gut health, improve digestion, and boost the immune system. Certain spore-forming bacteria are used as biofertilizers and biopesticides. For example, Bacillus thuringiensis produces spore and toxins that are effective against insects and pests, reducing the need for chemical pesticides and promoting sustainable agriculture. Spore-forming bacteria are used in the fermentation of various foods. Bacillus species, for instance, are involved in the production of traditional foods such as natto and certain types of fermented vegetables. Since spore-forming bacteria serve as model organisms in scientific research, their ability to form spores allow researchers to study cellular differentiation, stress responses, and the mechanisms of dormancy and germination. Right now, spore-forming bacteria are being explored as vehicles for vaccine delivery, since stability and ability to stimulate the immune system make them suitable candidates for developing oral vaccines that do not require refrigeration, which is particularly beneficial in developing countries. And who knows what more we can explore about bacterial spores? Maybe you could be the next Dr. Robert Koch who shaped the world of microbiology that we know today.